CRISPR is a biological gene editing system first discovered inside the genomes of E. coli in 1987. The purpose of this biological system is to cut viral DNA and store it inside the bacterial genome for use in the future. The storage of viral DNA allows the bacteria to remember how to efficiently fight off the viruses and thus become immune to them. This technology was studied by a few groups of scientists over the years, but not widely talked about until 2012. In 2012, Emmanuel Charpentier and Jennifer Doudna discovered that this bacterial immune response could be used to modify the genomes of other organisms by cutting the DNA in specific locations and creating desired mutations that serve beneficial purposes. What does CRISPR stand for? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. The CRISPR technology is comprised of both palindromic repeats and spacers. Each palindromic repeat, meaning that it is the same both forwards and backwards, is approximately 30 base pairs long and is separated by a spacer approximately 36 base pairs long. Each time a new virus enters the bacteria creates a new spacer for it, which is added to the CRISPR chain. The spacer DNA matches identically to the viral DNA so that the bacteria can remember the virus. This bacterial immune response has been adapted into a precise and efficient gene editing technology. It is comprised of two main components, the gRNA, guide RNA, and the Cas enzyme. In the gene editing process, the guide RNA joins with the Cas enzyme to form a complex. This means that they are bonded together. Inside the organism, the guide RNA is responsible for locating a desired spot in the genome, while the Cas enzyme is responsible for making a double-stranded break through the DNA. This cut in the DNA leads to an incorrect base pair configuration. This can knock out an entire gene or make minimal changes depending on the situation. Other methods can be used to add or modify genes as well. The guide RNA looks through the genome for a PAM sequence, proto-adjacent motif sequence, spelled PAM. The PAM sequence is typically 2 to 5 base pairs long and located 3 to 4 nucleotides downstream from the intended cut site. Only after the guide RNA has found a PAM sequence will it attempt to compare itself to the nucleotides following the PAM sequence. If the nucleotides are corresponding to the ones on the guide RNA, the Cas enzyme will activate and initiate a cut through the DNA. The problem is that not all genes contain a PAM sequence. This makes them uneditable to most forms of CRISPR. To get around this, scientists have been developing technologies that don't require a PAM sequence. By developing CRISPR technologies that don't require a PAM sequence to be present, scientists could edit any gene through the genome of any organism on the entire planet. This would give us unlimited possibilities for curing disease, producing biological products, and expand humanity's knowledge on genetics. This technology is incredibly cheap to produce and could revolutionize our planet. Treatments for cancer, HIV, and genetic blindness, among other ailments, have already been developed using CRISPR and are currently in the clinical trial phases. Besides curing illnesses, gene editing can also be used to make food more nutritious, develop garbage-eating microorganisms, and make organisms resistant to certain pathogens. This technology has only been around for 10 years, and the research and good news just keeps growing. I've explained what CRISPR is, what it can be used for, and why it is important. Now that you all know what it is, you may be wondering how to actually edit the genome of an organism. I've talked about the guide RNA, the Cas proteins, you get that, but then what? To help visualize the gene editing process, I did an experiment in the confines of my very own house for around $50. In the experiment that I performed, I edited the genome of E. coli. The goal of my experiment was to create E. coli resistant to the streptomycin antibiotic. To perform this experiment, you will need the following materials: LB agar media, which is a food source for the E. coli, LB streptomycin, kanamycin, arabinose agar media, which is food for the E. coli containing antibiotics, which will let us know if our CRISPR experiment has worked properly, a glass jar, a measuring tube, microcentrifuge tubes containing LB media, disposable transfer pipettes or reusable, sterile nitrile gloves, non-pathogenic E. coli bacteria, Cas9 guide RNA plasmids, bacterial transformation mixes containing polyethylene glycol 8000 and calcium chloride, sterile inoculation loops, and petri dishes. The first step of this experiment is to produce the food for your E. coli. This will involve mixing the required amounts of water and LB agar media together in a glass tube, and then microwaving them to dissolve the LB media. Once your LB media is thoroughly dissolved in the water, pour it into the petri dishes, ensuring that there's just enough to fill up the bottom of the petri dish. You will need to create both agar plates and antibiotic agar plates. This may get confusing, and if you mix up the plates, the experiment won't work, so it's important that you label your plates agar plates and antibiotic agar plates. Once you have your plates made, put the cover back on and let them sit overnight so that the condensation in the agar can evaporate. Once most of the condensation has evaporated, flip them upside down and store them in the fridge so that the condensation doesn't drip back down onto the agar. Now that you have your plates made, you'll need to prepare the E. coli. To do this, take it out of the freezer and add roughly 4 drops of sterile water. Next, you'll need to mix up the E. coli, and you can do this one of 2 ways. You can either use a sterile inoculation loop to stir up the E. coli, or you can use a pipette to suck up the E. coli and drop it back into the tube. Next, you'll need to streak your E. coli onto an LB agar plate. Suck up roughly 4 drops of E. coli into a disposable pipette and drop it onto the agar plate. Once dropped onto the plate, use a sterile inoculation loop to streak it across the plate in a zigzag pattern. This will evenly distribute your E. coli and increase your chances of a successful experiment. This next step will require waiting for a few days, but it is very important. You'll need to incubate your E. coli for them to grow properly. This can either be done at room temperature or at temperatures up to 37 degrees Celsius. Growing your E. coli at room temperature will cause them to grow slower, but it will still work. Growing your E. coli at temperatures around 36 to 37 degrees Celsius will allow them to grow faster and reduce the chance of mold or other contaminants growing alongside with your E. coli. E. coli grow optimally around 37 degrees Celsius, as this is the temperature that they're used to living in the digestive tracts of certain mammals, especially humans. Depending on the moisture levels and temperature that you grow your E. coli at, expect to wait between 2 to 6 days for enough E. coli growth to occur for the next step. In the beginning of this next step, your E. coli will start to grow. At first, you'll notice small whitish dots start to appear. As the E. coli ages, these small dots called colonies will get slightly larger and eventually turn yellow. All this means is that the E. coli is going through its life cycle and starting to mature. It's nothing to be worried about. In this picture, you can see a fuzzy green blob. It is a colony of mold that has started to grow due to me growing my E. coli around room temperature and having the petri dish in higher humidity conditions. To prevent this, simply grow your E. coli in low humidity conditions with higher temperatures around 30 to 37 degrees Celsius. Once you have fully formed colonies of bacteria, use a sterile inoculation loop to scoop them up and drop them into your bacterial transformation mix. This tube is going to be known as our competent cell mixture. This means that the E. coli cells can now take up plasmids and other nucleic acids which they normally couldn't. For now, we're going to put our competent cell mixture off to the side and take out a tube labeled plasmid. We're going to add roughly 2 drops of sterile water and shake the contents for 1 minute. Once we fully incorporate our plasmid into the water, we're going to use a pipette to bring it over to our competent cell mixture. This next process is called a heat shock. Our bacteria is mixed with chemicals, so after shocking it with a warm temperature, the pores of the cell membranes will fully open. We'll need to store our bacteria in the fridge at 4 degrees Celsius for 30 minutes to cool it down, and then immediately place it in warm 42 degrees Celsius water for 30 seconds before removing. Use a pipette and fill one of the LB media microcentrifuge tubes almost full with roughly room temperature water. After this, shake the tube to dissolve the LB media. Now, add roughly half of this LB media solution to your competent cell mixture and gently mix it by flicking the side of the tube. This next step is probably the most important step of the entire experiment. Optimally, you're going to want to incubate this tube at around 37 degrees Celsius for a minimum of 2 to 4 hours. In this 2 to 4 hour period, the bacteria will recover and replicate the plasmids that we've inserted into them. Once they have enough of the plasmids produced, they'll start to produce Cas9 enzymes and guide RNAs that will later form complexes and make the required mutation that we need. For my experiment, I let my E. coli sit out overnight for roughly 12 to 14 hours. This gives the E. coli more time to recover, replicate the plasmids, and produce the necessary materials that they need. After adequately incubating your E. coli, you can use a pipette to suck up roughly 4 to 5 drops of transformation mix and then drop it onto one of your LB antibiotic plates. Now we're just going to repeat the steps of the experiment that were done at the beginning to grow our E. coli. We're going to use a sterile inoculation loop to spread the E. coli across the LB agar, and then we're going to flip our plate upside down to prevent condensation from forming and dripping onto our bacteria. Then we're going to incubate it at room temperature for 4 to 6 days, or optimally at 37 degrees for 1 to 3 days. Once you see growth on the antibiotic plate, you know that your experiment was successful. If after a few days there's no growth, then unfortunately your experiment hasn't worked and you can try again. After doing this CRISPR experiment on the E. coli, I did almost an identical experiment on the same strain. This second experiment I didn't involve the CRISPR technology, but all of the steps leading up to the very end were identical. In this second experiment, instead of introducing plasmids that produce Cas enzymes and guide RNA into the E. coli, I introduced a plasmid that had a gene for producing the GFP protein, which stands for green fluorescing protein. This gene is derived from the Acoria victoria jellyfish, which naturally produces the GFP protein. By giving the E. coli the ability to produce the GFP protein, they will fluoresce under UV light, just like the Acoria victoria jellyfish.